So the last thing we're going to do with viruses in Lecture 6 is look through how influenza virus works. So we're going to look at how it replicates. It's usually for the best to look at a specific example to allow us to kind of put the generalities in place. So if you're having a little bit of trouble following my wordy slides about negative sense single-stranded RNA viruses, here is one, and we will see in detail what it does, and pictures will be worth lots of words. What's going to happen after this is we will learn about prions, which are totally different and terrifying. So this is probably the second to last video, and then prions, and then somewhere, if I have time, I will make another video about how the SARS-CoV-2 virus replicates in our host cells. This is not where I'm going to tell you what influenza is like as a disease. I'm not going to describe pandemics. I'm not going to talk about our immunity to it or vaccines. That's coming in Unit 4. And I'm going to do COVID in Unit 4 also. Here, we're really trying to understand what viruses are and how they work. This is our only really chance to dig into viral biology, and I desperately want my students to know what viruses are when they leave my class. So here we go. Influenza is a disease caused by the influenza virus. It's contagious and acute. It does spread in outbreaks. Each different government has its own way of defining what an epidemic is. So sometimes there are epidemics, sometimes there aren't. It's a member of the Orthomyxoviridae family. It is enveloped helical and negative sense single-stranded RNA genome. What we're going to see is it has a segmented genome. There are 8 genome segments, and each one has its own capsid. And that's why we're going to, well, I'll show you. The 3 major groups of influenza viruses, the ones that cause the pandemics that we think about, like that we're very afraid of a lot of the time, are influenza A viruses. B viruses cause seasonal outbreaks. And so if you get the trivalent influenza vaccine, it's usually 2 A strains and 1 B strain. C viruses don't seem to cause serious infections, so we don't think about them very much. Okay, so what is in an influenza virus virion? As we expect, there's negative sense RNA, so it doesn't directly encode anything. We would need to have its reverse complement to actually encode genes. It's segmented, so there are 8 segments, and what that means is if 2 different influenza viruses infect the same cell, like imagine a bird flu virus and a human flu virus infect the same cell, they can swap those segments. You could think of them like our own chromosomes, where we have 23 different chromosomes. They have 8 different genome segments. And that is a thing we'll talk about later. Each segment is part of a nucleocapsid, and that is proteins that protect each segment. There are 2 spike proteins, the H and the N. H is hemagglutinin, which is the receptor for salic acids, and the neuraminidase, which is an enzyme that degrades mucin, because this influenza is going to infect our ciliated epithelial cells in our upper respiratory tract, and so it has to get through mucus. So neuraminidase helps with that, and neuraminidase is going to help it during release. Influenza virions have an envelope, and they have a protein matrix that is like a big capsid right next to the envelope. So here is an influenza virion in a cute little cartoon. They've drawn the 2 different spikes, H and N, and you might remember H1N1, H5N1. Those numbers refer to the type of these 2 spike proteins. And then this is the This is the envelope, which was stolen from a host cell. And inside this is the matrix. We haven't talked much about matrix, but it's just a protein shell that protects everything on the inside, and it gives a nice anchoring point for these spike proteins. And then inside of that, in the core, so to speak, are the 8 genome segments, each one wrapped up in its own set of proteins. And I'll show you what these are like in more detail later. This is the big diagram we're going to look at to understand influenza virus replication, and I'll show you it bit by bit. So the more you look at it, the more you'll get it. The way textbooks tend to draw these processes is they draw the first steps over here, and then they draw later steps here, and the last steps over here, and then it's the end. So this is attachment, penetration and uncoding, biosynthesis, maturation, release. That's what's going on. So first we look at attachment. This is where neuraminidase is going to help get through the mucus. The hemagglutinin is going to actually bind the surface receptors, and the host cell is going to ingest this whole thing through receptor-mediated endocytosis. That will put this inside an endosome, or inside a vesicle. Remember, whenever we bring something in through endocytosis, it's inside a vesicle. One of the things we do is we acidify the inside of those vesicles. And so once that starts to happen, one of the proteins in the envelope that we haven't discussed, the M2 proton channel, it allows protons from that acidifying endosome to get into the core. And what that does is acidify the inside, and that helps separate the nucleocapsids from the matrix. So that helps, if we go back, let's see, that helps these separate from this. And that's going to let the virus uncoat. It's going to release the nucleocapsids. In this case, uncoding doesn't involve like removing. In this case, uncoding is mainly getting rid of the matrix. That's mostly what we're paying attention to, and that releases nucleocapsids. This is a nucleocapsid. This is a RNA genome segment with all the stuff that's attached to it. So what is attached to it? Well, the nucleoprotein is most of it. This is a capsomere protein that protects the RNA. That's its job. This is a negative sense single-strand RNA virus. So this is negative sense RNA. It cannot be translated. It must be a positive sense copy must be made, or a positive sense reverse complement must be made before anything else can happen. And that's what these enzymes are going to do. So these are 3 different enzymes that work together to do all the different little steps that have to happen in order to start making a reverse complement. So that's PB1, PB2, and PA. Those are different polymerase basic, number 2, polymerase basic, number 1, polymerase acidic. I don't know basic, acidic, I don't know the details there, but the 3 of them work together to do all of the nucleic acid synthesis that it does. Biosynthesis is where we expect this to be kind of gnarly because it's a negative sense single-stranded virus. So buckle up, we're doing this, and it's not as bad as you think. Maybe I'm the only one who thinks it's bad. I'm the only one who's scared of this. You're a very knowledgeable cell biology person. Okay, so this is viral RNA. This is the RNA that came in as part of a nucleocapsid. So this was in the virion, or this is what is going to be put into a new virion. So this is the RNA that goes in a virion. So it's negative sense, what do you call it? Negative sense single-stranded RNA. In order to do any translation, we have to make a reverse complement, or influenza virus has to make a reverse complement. So it uses these little enzymes to make a reverse complement, and that ends up acting as messenger RNA that is sent out of the nucleus into the cytoplasm. How did all this get into the nucleus? I don't know how that works. I don't know. It's complicated. I have no idea how anything gets into a human cell nucleus. Someday when I'm older, I will read all about that. Anyway, messenger RNA gets pushed out of the nucleus where it can be grabbed by ribosomes. Some ribosomes are free-floating in the cytoplasm and others are attached to the endoplasmic reticulum. And the ones that are in the cytoplasm are going to make cytoplasmic proteins that can just hang out in the cytoplasm. The ones attached to the endoplasmic reticulum are going to put enzymes in the membrane of the cytoplasmic reticulum. The endoplasmic reticulum, sorry. And then these are ultimately going to be sent to the Golgi in vesicles. And so all of these are destined for the cell membrane, or at least vesicles. So that was what we've talked about. These enzymes make the reverse complement, which is messenger RNA, and that can lead to synthesis of all these proteins. But the virus also wants more viral RNA. It wants to make lots of copies of itself. So it needs to make lots of copies of this molecule. To do that, it has to make a complementary strand like messenger RNA, but it doesn't actually use this to make the copies. It makes what we call a copy RNA, which is the same as this, except that it doesn't get used as messenger RNA. It only exists as a template for making more of this. So this is the reverse complement that allows the virus to make many copies. So at this point, you should be able to explain where every object here came from. Not what it's going to do or what its purpose is, but you should be able to explain where every part of this came from and how it got where it is. So I've just gone through everything on here and how it got where it is. So by the end of biosynthesis, all of these viral proteins are in the cytoplasm. All these viral proteins are in the endoplasmic reticulum, and there's viral RNA being made in huge quantities in the nucleus. This is a critical thing that many different viruses have to do. It turns out messenger RNAs need to have 5' caps, and also in order to make reverse complements, they have to have a primer. They have to have a first base laid down that they can use to add the bases to as they make a new nucleic acid. And they do all of this together. So what you're looking at here, is this is a nucleocapsid. So this is a genome segment with the 3 different polymerase enzymes attached to it and the protective enzymes on the RNA. And this is a cellular messenger RNA. This is one of our RNAs with a 5' cap and a poly A tail. Well, influenza has the enzymatic activity to steal the 5' cap from one of our RNAs and use it as a primer to start its own synthesis of the its own RNA. So that's how it makes the viral RNA, and it has a 5' cap that it stole from this cellular messenger RNA. This is really common in viruses. Lots of them do what we call cap snatching. And this is just one example I wanted to give you of the things I haven't really talked about, of the more complicated things viruses have to do. And the more you learn about viruses, the more you have to learn more and more and more about viruses to understand what's going on. Right, so influenza steals the cap. It's cool. It's clever. it sucks for the host cell, because remember what the 5' cap does is it makes the messenger RNA stable and it helps it get translated. And now this messenger RNA is not stable and it will not get translated. So that was all part of biosynthesis. So cap snatching was part of biosynthesis, and all of this transcription, translation, RNA copying, that was biosynthesis. Now we look at maturation and assembly. So there are two kind of pathways, two kind of things happening here. One of them is all of this stuff. Well, what do we have here? We have NP, we've seen that before, that's the nucleoprotein. We have basic polymerase 1, polymerase basic 2, polymerase acidic. These are part of the nucleocapsid. So these are all going to get trafficked back into the nucleus where they're going to associate with the viral RNA. Fair enough. So they're made in the cytoplasm because that's where the host makes proteins. And then they get sent somehow, I don't really know how that works, back to the nucleus. Two other proteins, the matrix protein and another protein, I'm not sure what this one does, they're going to get attached to each genome segment. And these are all going to be attached to a vesicle of some kind that is going to be sent to the cell membrane. And so this is a process that is kind of outside of basic cell biology. I'm not sure what these things are hijacking to make this happen, but ultimately they assemble the nucleocapsids here with all the enzymes that have to be attached to it and the matrix protein, and then they send those to the cell membrane. And so the matrix proteins are going to start making this spherical shape, and they're going to start attaching to the nucleocapsids. And think about how tricky this is. There are 8 different genome segments, and it needs exactly one of each. So it has to somehow be able to figure out which ones it has. And this is happening all over the cell. There might be 50 different versions of this happening at the same time. And they all have to figure out exactly which segments go to which virion. Up here, you're seeing the normal protein trafficking for membrane-bound proteins. You're seeing the endoplasmic reticulum is where they're made, and through vesicles they're sent to the Golgi, they're organized in the Golgi, and through vesicles they are sent to the cell membrane. And so that's the spike proteins we're seeing here. That's neuraminidase and hemagglutinin. And then the M2 proton channel would be put somewhere in the membrane here. So that's maturation and assembly. After you work through this, I don't expect you to understand exactly how this works, but the end product is keep track of what these proteins do and where they go. And then this you should understand, because this is how normal host proteins get to the membrane through the endoplasmic reticulum and the Golgi. And then the last thing is release. This matrix makes a full sphere, and this part of the membrane touches this part, and the virion is free. So it's a lot of stuff to work through if you're rusty on your cell biology, but it's elegant, and what it does is give us a window into kind of how our own cells work, and the fact that there are extra proteins here, like why are there 3 polymerases? Why is there this extra protein? That gives us a sense of what other challenges the virus might have to be solving. A lot of times viruses will have genes, we have no idea what they do. So they'll be making some protein, we don't know what its function is. We know it has to solve some problem in the cell, we don't know what the problem is. We don't know how the solution works. But the reason I use influenza as an example is because it's one of the most important viruses we could possibly study, and so this is really well known. Even though I don't know how it all works, people do understand influenza very well. what I want to leave you with at the end of this video is something that you should be able to fill in. It's comparing bacteria with viruses. What is the difference? I want you to be able to tell people the difference. I want you to think about what you've been taught, because I've noticed that LCC students have been taught things like, I don't know, my previous students had been taught some real nonsense about the difference between bacteria and viruses. You should be able to come up with this list. So, are they obligate intracellular parasites? Some bacteria are. We haven't really talked about them. All viruses need cells to be made. Do they have plasma membranes? Bacteria have to, right? They have to separate the inside from the outside. Viruses only do if they need an envelope. They don't really need to do, they don't do electron transport. They don't move proteins in and out. They like wear a membrane instead of needing to use a membrane. Do they divide by binary fission? Bacteria do, viruses don't. Viruses are assembled at full size. In the laboratory, can we remove them from water using a filter? Yes, we can. Bacteria are big enough we can filter them out. We really can't filter out viruses using typical laboratory filters. If we were to look inside them, would we find both DNA and RNA? Well, if we look inside a bacterium, yes, we're going to find DNA. We'll find messenger RNA, transfer RNA, ribosomal RNA. In a virus, we're going to find one or the other in the virion. Do they generate ATP? Bacteria have to. Viruses don't. And do they have their own ribosomes? Bacteria have to. Viruses don't. So really, I guess I should clarify, we're comparing virions with bacteria. Why do we care about virions? Well, these are the things that get us sick, right? This is what you're avoiding. If someone around you has COVID and they're sneezing, you're trying to stay away from the virions they are releasing. So I want you to be able to explain the difference between those virions and a real cellular life form. Hopefully by the time you work through these slides, this will be an easy thing for you to do, and you really will have a sense that they are very different. That's my hope. If I'm wrong about that, tell me in office hours. Okay, so that's it for viruses. There's one more video coming, and it's about prions, as you will see. So I will see you there.